tell me, did you guys physics class was going on so late? Yeah. I understand. I get it. No problem. So guys, tell me, like, you if you have any doubts, please let me know. So that's first thing. In the last class, I have given you some of the work, not work. Basically, it was for yourself to practice the questions. I hope you went through the questions. If you would have gone through those questions that I sent you in the form of a PDF, so it doesn't matter what source it is from, it is the type of questions we are going to address, correct? No? So that particular paper contained a lot of questions on pedigree analysis and genetic diseases, correct, right? Inherit the disease patterns or something like that. So if it have gone through each of them, even the wrong ones or something like that, it would have benefited you a lot, and you would have a solid understanding of the genetics and how it works. And I hope some of you would have worked out. Okay, hopefully. Chalo. So today what we do is good. We have a very good strength. Today we start from pedigree analysis. And the last class I gave you an introduction to what is pedigree analysis. So by definition, what we do is we do a symbolic representation of generations. For example, let's say we have great grandparents until where we can trace and where the memory lasts. So we take those and we start plotting against one generation after another generation. So why do we do this? In normal cases, it does not give us much of the output or evidence. But if you target a specific character, if you target a specific disease, if you target a specific genetic trait, then it would become very easy on what we are looking at, correct? No? So when we look at it, then when we go from one generation to another generation, so we understand the nature of how the gene is getting expressed. For example, if you look at the Mendelian genetics and how the Mendel started expressing his work, when he performed a monohybrid cross and then he took a tall plant and a dwarf plant, in the F1 generation, was he able to tell us what is dominance, what is segregation, what is a law of paired factors? So was it possible for him to predict it? Definitely no, right? So he had to go from F1 generation to F2 generation. So when he went from F1 generation to F2 generation, then what happened? He literally understood that, okay, for me to able to understand why the dwarfness appeared in the F2 generation, so this is the possibility, correct, right? Then he understood, okay, this gene should be dominant, this gene should be recessive, they should be present in pairs, and they segregate independently of each other, etc. So similarly, when we trace any genetic character from one generation to another generation, we get a very clear idea on how it is getting expressed. And the best way to do it is through pedigree analysis. So here is a chart. So the chart gives us a idea on how the symbols are used and how it is going from one generation to another generation. Let's see here. So if it is a square, it is a male. If it's a circle, it's a female. If a square and a circle with the link, it is called as mating or married. Okay. Next one is a square, a circle, and then the next generation comes with one square and a circle. You know that the parents are having a one girl and a one boy child. Okay. Next, whenever you have the next generation and it is given within the triangle, okay, when the triangle shape is represented, that means we are talking about dizygotic twins. So what's the difference between dizygotic twins and monozygotic twins? Why is it even significant for us to differentiate between them? So you know that monozygotic twins characteristics are absolutely similar. I mean, like, there is high amount of similarities between them. Literally, when they stand together, you won't be able to identify who is who. That is in monozygotic twins. When the kids are born from the same zygote, when a zygote breaks and then embryo breaks basically, and then it becomes two babies. Same embryo breaks into two and the two becomes two babies. That is called as monozygotic twins. In dizygotic twins, there are chances that these twins have differential characteristics. Even though so they share a major amount of similarities, but you can differentiate clearly between who is A and who is B. Okay, so that is dizygotic twins represented by a triangle. Okay, now if you have a monozygotic twins, you see in monozygotic twins also there is a triangle, but you can see that the sex of a baby is same. Okay, the sex of a baby is same. That is square and square. or round and round, that would be a monozygotic twins. So a rhombus would be sex unspecified. Okay. And then if you have a square and the numbers are written in between them, within them, like 2 or 3, so then you can see the number of children's of sex indicated. For example, within the square there is 2, that means there are 2 children, and there is circle and there is 3, that means there are 3 children. Then comes the important part. The important part is when it is shaded. Male square is shaded and the circle is shaded, that is called as affected individuals. And if they are half shaded, that means they are the carriers of the diseases. So usually whenever we have a half shaded ones, they are heterozygous in nature. Remember this. They are half shaded ones are heterozygous in nature, and they are usually for autosomal recessive diseases, they are heterozygous. Correct, no? Next one is you have a dot, you have a circle, and within the circle there is a black dot. Okay, that is called as carrier of sex-linked recessive disorders. So usually you can see that in females. In female are the ones who carry the sex-linked recessive disorder, correct? No. For example, you can take an example of hemophilia. In hemophilia, you can see that the female is a carrier of the disease, and she has this defective chromosome on the X. So that is an example. So here you can see that there is a black dot within that. So she is called as a carrier. Next one, if the baby is given in a square, okay, and if the square is struck off, that means the baby died. Okay, the boy baby died or girl baby died. Okay, that means death. Next one is if you have a line and then if you have a circle, that is called as stillbirth or sex unspecified. Okay, so anyways, these are some of the symbols that we use for the representation of pedigree analysis. Now you can see that we can really go deeply into their behavior, their genetics, their characteristics and everything, and represent it over a generation over generation. And then we finally can conclude what can the next generations be, what are the possibilities, what was the reason in the past, how is the present, and how would be the future. So that's why pedigree analysis is way too important. Okay, now look at this chart. In this chart we have half shaded individuals. Now you can see wild type female means normal. Okay, and then you have wild type male, that's means normal. Heterozygous is half shaded. Again, heterozygous female is half shaded. Homozygous male means completely shaded. Then homozygous female means completely shaded. So this is the ones that we represent through pedigree chart. Anyways, let's go for the genetic disorders. So in genetic disorders, there are 2 types of disorders. One is called as Mendelian disorders, and the other part is called as a chromosomal disorders. Why do we have to differentiate between Mendelian disorders and chromosomal disorders? When it is a genetic disease, it's a genetic diseases. Again, why we have to go and split them into 2 more categories? The reason is very simple. Whenever we talk about a disorder, we talk about the behavior of the disorder, correct? No. So in order to understand the behavior of the disorders, we go through the pedigree analysis. So whenever a genetic disease or a disorder expresses in the form of Mendelian laws, for example, law of dominance, law of segregation, law of paired factors, or you see the law of independent assortment. So when they follow Mendelian genetics very strictly, those are called as Mendelian disorders. But you have some disorders which do not follow Mendelian disorders. So when they do not follow Mendelian disorders, that doesn't mean that these genetic disorders do not exist or they're very rare. No, it's not like that. They follow different patterns of expression. What is that? They follow chromosomal inheritance, bulk transfer of genes. For example, if there is one additional chromosome present in the diploid condition instead of 46, if a person has 47 chromosomes, if a person has 45 chromosomes, okay, so this shows the haplontic variation with respect to chromosomal number, which we call it as chromosomal aberrations. Okay, so whenever they come, you don't specify to one gene, you specify to a group of genes, and you target and you basically pinpoint on the defects of the chromosome rather than a single gene. So this is called as a chromosomal disorder. Okay, so let's see. If you see the examples for Mendelian disorders, okay, so usually it can be hemophilia, it can be color blindness, cystic fibrosis, sickle cell anemia, and then you have phenylketonuria, and then thalassemia. So these are the examples for Mendelian disorders. Chromosomal disorders examples would be Down syndrome, Klinefelter syndrome, and Turner syndrome are the textual examples that we usually study, okay, for these diseases. So let's go first one. First one is the hemophilia. So under chromosomal, under Mendelian disorders, the first disorder that we are going to target is hemophilia. Okay, before we talk about hemophilia, now my question for you is, what do you know about clotting factors and how many clotting factors are there? Can you please tell me? I will wait for your response. What are clotting factors and how many clotting factors do we have, children? Very beautiful. We have good strength today. Very nice. I like it. Gives me a lot of josh. What are clotting factors? And how many factors do we have? Don't Google. Just tell it. Just tell me. If you Google, I will ask you questions on what factor is what. Okay. You guys have an answer. Your generation should be very fast with computers, you know. We are slow, very slow. You can see me struggling with sometimes erasing. Even erasing, I feel struggles. Okay. So come on. What is a clotting factor? How many factors are there? Okay. Sorry. Yeah. How many factors are there? Can you please tell me faster? You can unmute. 13 clotting factors are there. Okay, 13 clotting factors. That's an interesting answer. Very interesting answer. Okay, usually in your textbook or any books you study, you come across 12 clotting factors, which is the 13th factor. So I told you, I'll ask you a question. Yes, yes, yes. Now somebody is arguing, is it 12? Somebody is telling 13. You see, the 12 is, these guys are correct. Who are those? Let me look at, one is, uh, I'm sorry, guys. I can't read the person. Oh, sorry. The person who told 12, I am really sorry. I'm not able to read the name. Ching, JNV, something like that. Okay, sorry. And the other person, the Sneha is telling it is 13. Okay, both are right. Here, both the guys are right. Basically, the clotting factors is 1 to 12. You understand this point? 1 to 12 is the clotting factors. Whereas there is a 13th factor, and the 13th factor is basically called as stabilizing factor. Okay, so what are we talking about? We are basically talking about, okay, yeah, perfect. We are basically talking about, we have factor 1, factor 2, factor 3, factor 4, factor 5, factor 6, all the way till 12. Now, why these clotting factors are even present at the first place? If you see the clotting mechanism of the blood, the clotting mechanism can be divided into 3 phases. Okay. First one is called as formation of platelet plug. For example, when I have a cut and I have a blood vessel, you see guys, this is my blood vessel. And when a blood vessel is split apart, so the collagen fibers that are exposed in the blood vessels, they activate our platelets. So the platelets are basically are very soft, and they become, with the exposure with respect to the collagen fibers of these blood vessels, they become spiky and sticky. When they become spiky and sticky, they start clumping with each other. When they start clumping with each other, they create a plug-like structure in the blood vessel so that they avoid the flow of blood. This is a temporary structure. Usually takes around 2 minutes, okay, or to 30 minutes. So here, what happens is it's not permanent. If you squeeze, again, the plug will get broken down and then the blood start flowing out. Okay, that is one thing. The second one is your you have your blood vessels, and in your blood vessels you have your circular muscles, and then you have longitudinal muscles. So what happens is, this is called as vascular spasm. Vascular spasm में क्या होता है? You have the diameter of a blood vessel, right? So this diameter of a blood vessels get reduced. Let us assume that this is the diameter of the blood vessels. It get reduced like this. First thing, the reduction of the diameter of the blood vessels takes place, and the second point is they get drawn away from the site of injury. That means the blood vessels get literally pulled apart from the site of injury so that the leakage is reduced. And the second point is the exposure of the blood to the outside environment is also avoided. So this is the first two basic steps that happens. So in both the steps, this is called as clotting. Okay, here it is temporary steps. And the third important step is called as coagulation mechanism. In coagulation mechanism, in coagulation mechanism, it is a very detailed mechanism and it is also called as a cascade pathway. So cascade pathway, okay, what is cascade pathway? Cascade pathway involves, like these guys told, 12 factors plus one factor. Sir, do they ask us how many clotting factors are there? Definitely no, guys. In detail, in reality, it's 13. Remember this. But in textbook, it is 12. So why are you confusing us? Is it 12 or 13? Guys, the one who participates in the coagulation is 12. The final one is the one who stabilizes the whole process. You understand this point, right? So if they ask what is the total number of clotting factors, it's 12. Remember this. So we have factor 1 to factor 12. So for example, if I take factor 1, the factor 1 is your fibrinogen, which converts finally into fibrin. Okay, fibrinogen, it's a fiber, okay? It converts into fibrin. So these fibrin, they come and they, make a stitches, stitches like a meshwork. And finally, they create a proper coagulant, okay? So that is how you make a permanent clotting of blood, coagulation of blood. So anyways, all the factors are important. But here, why am I talking about clotting factors? See, I told you that it is a cascade mechanism. Whenever in a cascade mechanism, all the factors are important. If even one factor is missing, then the coagulation mechanism completely stops or it does not happen at all. So we have factor 1 to factor 12, right? So in this, we have few factors. What are those? Factor 8, factor 9, and factor 11. Do you know these factors? What are these factors are called as? Factor 8, factor 9, and factor 11. These factors are called as anti-hemolytic factors. Okay. What are factor 8, factor 9, factor 11 are called as? They are called as anti-hemolytic factors. Okay. Factor 8, anti-hemolytic factor A. Factor 9, anti-hemolytic factor B. Factor 11, anti-hemolytic factor C. Hemophilia, hemolytic. Are you able to are you able to find a connection here? Definitely, right? So you can find that there is something that is kind of relevant here that we are talking. So what happens if anybody who has a genetic disorder where they cannot produce factor 8, they will have what? They will have hemophilia A. If they cannot produce factor 9, they will have what? Hemophilia B. If they cannot produce factor 11, what do they have? They will have hemophilia C. So what is happening here? During the coagulation mechanism, we have factor 8, factor 9, factor 11. If there is any disturbance in the production of factor 8 protein, factor 9 protein, or factor 11 protein, that leads to a genetic disorder called as hemophilia. So what is hemophilia? Hemophilia is also called as a clotting disorder. Okay, what is it called as? It is called as a clotting disorder. So hemophilia comes with different names. It is called as Royal's disease. Okay, it is called as a royal blood disease or Royal's disease. Okay, or it is also called as bleeder's disease, etc. Why bleeder's disease? I told you the permanent clotting of blood cannot be accomplished because one of the clotting factors is not produced effectively. So that is why these people have the problem. So how does the disease come into existence in the first place? So this particular hemophilia disorder comes because there is a gene that is present on the X chromosome. Okay, what gene? Anti-hemolytic gene. Okay, the anti-hemolytic gene is present on the X chromosome. What is the nature of the gene? When I talk about the nature of the gene, either it has to be a dominant gene or it has to be a recessive gene. Correct, no? So this particular gene is a recessive gene. So what is the meaning of recessive gene? That means in order for it to express, it has to be in what state? It has to be in the homozygous recessive state. That means both the chromosomes should carry the affected gene. So first thing, what did we understand? We understood that hemophilia is a recessive disorder and it is present. The gene for anti-hemolytic factor is present on which chromosome? X chromosome. You got my point. Since it is present on the X chromosome is what chromosome? It's a sex chromosome. That is why we call this hemophilia as sex-linked recessive disorder. Got it? If a gene is located on the sex chromosome, it is called as sex-linked. If a gene is present on the autosome, it is called as autosomal linked. Okay. If it is dominant, it is called as dominant gene. If it is recessive, it is called as recessive gene. Got it? No? So what happens here? Now, I told you how the disease is caused and what is it, how is it responsible. So let's see what is the chances. What are the chances that we can see in a hemophilia? So these are the different possibilities in which a people can marry. Okay, let's see. Here we have a man and we have a woman and they are marrying. Okay. Now the woman appears very normal. Whenever she gets cut in the hand, she doesn't get affected and she's totally normal. Okay, she's totally normal. Now what happens is when they have kids, one boy is normal, one girl is normal, other girl is also looking normal, but the boy, the one child, whenever he gets cut, he doesn't stop bleeding. What happened here? Now here, if you look at the pedigree chart, if you look at the crossing of how this happened, you can see here the female was in X dash X state and the male was normal. He was XY, correct? No? His chromosomes are X and Y. I hope you can see the letters I am writing on the board. If not, please put it on the chat box that it's not visible. We can do something. So what are the chances of them making kids? You can see here X dash X is one condition. So this X dash and this X, guys. Okay. Again, X dash Y. Then you have XX. And then you have XY. Correct, no? You know how to conduct a cross now. Now you understood that these are the possible ways we can get a kid. Now, sir, why didn't this X dash X condition may hemophilia did not get expressed? Okay, not visible, not visible. The upper portion is not visible. Okay, guys. Okay, no problem. Okay, no problem. What we do, I write here. I write here. Okay. Here. Okay. X dash. Okay. Sorry. Okay. Here I think I'll write. X dash X is a female. Okay. I hope now it is visible. She marries a man who is normal. Now their kids will be what, guys? That's what I was saying. Their kids will be X dash X, X dash Y, X X, X Y. Correct? No. So when they marry, these are the possible kids they can have. So now what is happening here? Okay, hold on. See you guys. Or sorry. Okay, fine. Now you will see if you have any problems. Now you see, you can ask me, sir, why dot X dash X is not showing any hemophilic conditions? See, the nature of the gene is it is in a recessive condition. So even though one chromosome has a defective gene, the other chromosome has a healthy gene. The X dash is defective, the other X is very healthy. And what is the nature of the gene? In the X gene or the hemophilic gene is a dominant gene. The defective hemophilic gene is a recessive gene. So in order for the recessive character to be exhibited, it has to be in a homozygous recessive state, especially in females. You understand this point? Now, this condition you understood. Why X dash X does not express? X dash X does not express because another X has a healthy gene and that gene is dominant. Simple and plain. Next one. Why in this case the male gets to suffer whereas the female does not get to suffer? She becomes a carrier of the disease. I'll tell you the meaning of carrier of the disease. Just give me a second. So here what is happening? X dash Y may. X dash Y may is expressing because there is no counter genes. That means what? The Y chromosome do not carry any gene for hemophilia or what do you tell? Yeah, hemolytic factors. It does not carry any gene for hemolytic factors. So since it is not responsible for production of that protein, okay, then Y cannot avoid this. So when Y cannot avoid this recessive gene gets expressed. So even in X dash Y condition, the male becomes the expressor. You understood my point? Okay, chalo. Now let's see here. This condition you understood. Now let us assume a man is the one who is an expressor of hemophilia. The woman had a great heart, so she will marry him anyways. Okay, she was very, very normal, fine woman. She has no genetic disorder. Now, what would be the possibilities? The possibility is all the male child that are born to these couple will be normal, and all the female that will be born for these couples would be the carrier of the disease. Now, I told you that I'll explain you the meaning of carrier of the disease. The carriers of the disease are the ones they carry the defective gene in one of their chromosomes, whereas the other chromosome has a healthy gene. You understand my point? What is a carrier? A carrier is the one who carries the defective gene in one of the chromosome, and the other chromosome has a healthy gene. Now, why do we have to give importance to them? We have to give importance to them because the carrying of defective genes is very, very, very secretive. Now, what is happening? Now, these are the ones who have the potential not to get recognized. Correct? No? When you look at this female, you can't tell that she is having a genetic disease. How do you go? You can't ask a pedigree chart. You can't ask for genetic chart from a horoscope. When you go marry, you can't do that. That's why they're hidden. You can't find out where is the disease. They are very, very healthy. They are very normal like you. So, okay, but unfortunately, they carry a defective gene. That's why in the next generations they get expressed. Okay, now I hope you know how this condition works. Okay, so how does this condition work? You look here. We have a defective X chromosome, that is X dash Y, and then you have XX. Correct? No, people. Now here, X and see, for a guy to be born, what should it be there? It should be X. Okay, this Y goes with this X, so it will be XY. Then X dash X. Then again, you have another X chromosome going with another Y. So you have XY. Then again, X dash will go with another one. So X dash X. So you have two defective females, and then you have a, two normal male. The females will be carrier. Okay. In what condition a female can be an expressor? It is extremely rare. It is an extremely, extremely rare condition. We have not seen such conditions. Maybe it is possible. We can see such conditions, but it is absolutely rare to have a defective female who is expressive. Why? What is the requirement for it? Imagine the chances. The chances are the male should be the expressor of the disease. The female should be the carrier of the disease. Already she should have a defective chromosomes. That means what are we talking about? X dash Y should marry X dash X. So this is a very, very rarity in our nature. So what is happening here, guys? You can see here the male is normal. Only one male child will be normal. The rest of them will be defective. How? One female will be expressor. This is the rarest individual ever. She'll be hemophilic. Next one, another male child will be effective. He'll also be hemophilic. Whereas another female will be a carrier. I will not work out how this cross works because you guys already know how it works. We have come this far. Please try to do this. Now, why did I explain so much? I explained you so much because the rest of the topics should be quite easy for us. Did you understand anything? Okay, all good? Do you guys have any doubts? Okay. Oh, you guys are giving thumbs up. I never saw thumbs up. No, okay, fine. Okay. Other guys, please try to, what do you say? Communicate as well. Okay. We have less classes, guys. I need to know you because when I meet you, I don't want to meet strangers. Okay. Clear? So, chalo. So let's go for the next one. That is color blindness. See, color blindness is also a sad disease. Of course, it is sad because what is color blindness? Color blindness is an inability to differentiate the pigments. So usually what happens whenever you see colors, the colors are, you have basic colors in the form of pixels. So each pixel contains the 4 colors. One is the red, yellow, green, and blue, I feel. Yeah, correct. So these are the 4 colors. So the combination of these colors would give you whatever the shades that you see, like the blue, light blue, white, and everything. Okay. So that is how it works. Okay. Now, how are we able to differentiate colors? we have a special proteins that are produced in our rods and cones. Okay, so usually what they do is they absorb a specific wavelength and they are very photosensitive to this wavelength, and they create an optic signal. Through this optic signal, you generate an image of that specific color. Now, if this protein is not produced, then you have a color blindness. So let's look at what is color blindness. Color blindness is basically X-linked. Again, X-linked means it is a sex-linked disease. What kind of disease it is? It is a recessive disease. That means when I say it is X-linked recessive disease, they have the same pattern of inheritance as that of hemophilia. A female becomes a carrier, a male becomes an expressor. A female becoming a colorblind is extremely rare. When can a female become a colorblind? When she marries a man who is expressing the colorblind, okay, and her mother should be a carrier of the colorblind. Correct? No, we just saw. The only case in which a female baby can be a colorblind is when the parents, one is expressor, other one is a carrier. So only then they can be colorblind. Okay. So what is happening here? We have rod cells, okay, which are active in low light, and then we have cone cells, which are usually responsive in the bright light. So they are responsible for color perception. Okay, so there are three kinds of cones. So each are sensitive for a specific wavelengths. So you can see here in our eye, we have 64% of red cones and 32% of green cones and then 2% of blue cones. Okay, guys. Okay, so you can see here we have red color blindness, we have blue color blindness, and we have complete color blindness. Okay, if we have red color blindness, okay, it is called as protanopia. Okay, protanopia is red color blindness, tritanopia is blue color blindness, and then you have monochromatism. That means you cannot differentiate any colors. Okay, and then you have your deuteraphia. Deuteranopia. So deuteranopia is green color blindness. Okay, protanopia is red color blindness. Tritanopia is blue color blindness. If no color is differentiated, it is called as monochromatism. You get my point, guys? So I'm sorry, I cannot ask you the question. Can you see all the colors here? So that depends. Okay. Anyways, so you can see that in monochromatism, what happens is it's called as a complete color blindness condition, okay, in which all the colors will appear as light gray, dark gray, or medium gray. All the colors, okay, it's like a black and white. You get my point? And then you have the dichromatic condition. In dichromatic condition, it is called as a partial color blindness condition, where only two color perception is happening. Either you have the red and blue or blue and green, okay, any of the combinations will be there, okay. So next one is you have daltonism. In this condition, what happens? He'll be unable to identify only red and green color. Like that, I gave you the definition for what is protanopia, and then you have your deuterophia. Okay, so let's see the genetics of this particular color blindness. See, color blindness ka genetics works exactly like that of your sickle cell anemia. Imagine that we are just plotting a chart of color blindness instead of hemophilia. You understand my point? So the same way it starts expressing. If you have any doubts, you can ask me right now. Sir, how does it express? Same, guys. It is an X-linked recessive disorder. So this disease is caused because you have a defective gene on the X chromosome. So the defect of the gene can be of many types. It can be all the three proteins are not produced, one protein is not produced, or two proteins are not produced. You understand what I'm telling when I tell proteins? So the protein that is present in the cone is absent. When the protein is present in the cone is absent, I mean, like when the protein is absent in the cones, then you lack the ability to differentiate a color. Correct? No? So when you lack the ability to differentiate a color, okay, hold on. You want the previous slide? Okay. This is a previous. This one is a previous slide. You want this one or this one? Okay, sorry. Yeah, this is the slide I feel. Okay. Are you understanding, guys? Simple, no? So knowing one would give you the ability to understand others. Only thing that you have to understand is what type of disease it is. You'll forget. Okay. So it is sex-linked recessive disorder. When we talk about sex-linked, it is X-linked, that is present on the X chromosome. What is present on the X chromosome? The gene responsible for production of proteins in the cones that help us differentiate colors is absent, or it is defective in the X chromosome. That leads to different types of color blindness. So usually they have asked you, what is this deuterapia, what is protanopia, what is tritanopia, and what is monochromatism. They have asked in examinations for sure. They have asked. I've seen some questions coming from, okay, tritanopia. I've seen questions from asking from what is monochromatism. Complete color blindness is also called as what. So these questions have been asked. Okay, guys, you understand me? What I'm trying to say? Okay, so let's go for the next one. I hope Jabra Sultana, you're okay with it, and if you're not okay, please let me know. Let's go for the next one, that is sickle cell anemia. So in sickle cell anemia is an interesting case. Okay, so what is happening in sickle cell anemia? What kind of disease it is, guys? What kind of a disease it is? Is it autosomal linked recessive, or is it sex linked recessive? Can somebody tell me? Okay, so while you type your answer, because you have heard this lesson, so let me ask you a question. Is it sex linked recessive, or is it autosomal recessive disorder? So what would be the situation here? So as you guys type down the answers, let me go and explain this topic. So sickle cell anemia, what is happening here? You have to understand that our red blood cells are very well designed to pick up, they're like a carriers of oxygen molecules. And they do this carrying of oxygen molecules with the help of a protein called as hemoglobin. Okay, in hemoglobin, okay, so hemoglobin is a protein and it is a globular protein. So the shape of a hemoglobin protein is globular in nature. So what is the meaning of globular in nature? So that means protein is a structural molecule. That means its action or its capacity to function majorly depends on the structure it has. So structure of a protein becomes a very core characteristics of how the protein can function or whether it can function or not. So if I ask you what is a shape or what is the shape of a protein or what is the shape of hemoglobin, you should tell that it is a globular protein. What is the meaning of globular proteins? Basically, we can differentiate proteins into primary structure, secondary structure, tertiary structure, and then you have quaternary structures. So we'll study this, okay? In quaternary structures, you have globular proteins. So that means what? They are made up of many types of polypeptide chains. If you look at the hemoglobin, let us say that this is the heme structure, that is the iron. Okay, the iron is in the center, heme. And then it has four chains, 1, 2, 3, and 4 chains. In these 4 chains, you have 2 identical chains called as alpha alpha chain, and then you have 2 more called as beta beta chains. So this is called as an alpha globulin chain. This is called as beta globulin chain. So how many you have? You have 2 alpha globulin chains and you have 2 beta globulin chains. So whenever you define sickle cell anemia, you in your textbook, you are very much aware now. In the 6th position of a beta globulin chain, okay, glutamic acid is replaced by valine. So that leads to sickle cell anemic state. Correct? No? You would have bioted back then. So that is what is exactly happening. You should understand what are we talking when we talk about beta globulin chain. So hemoglobin is a globular structure. A globular structure will have many peptide chains. Each peptide, when I say a peptide chain will have what? A sequence of amino acids arranged in a certain fashion. So we have 4 chains. They have their own particular way in which the amino acid sequences are present. So these 4 chains, okay, are called as what? Globular chains or globulin chains. So what are the globulin chains that hemoglobin have? Alpha alpha, beta beta. So where is the effect that we can see? Whenever we see the protein, okay, formation of a protein, the formation of a protein is a direct way in which you can understand how the gene expresses or how the gene functions. Correct? No. So what does DNA have? DNA have genetic codes. That means you have A, T, C, G, G, like this. The codes are arranged. Why are these codes are arranged? The codes tell us what amino acids should be arranged next. Similarly, you have the list of amino acids that are arranged. You can see here in the beta chain, you have CAC, GTG, GSE, TGA, and then you have GGA, and then you have CTC, and then you have CTC. You get my point? Okay, this is how the sequence of amino acids that are arranged in your hemoglobin molecule. In a normal case, see, this is the first, second, third, four, five, six. This is the sixth position. In which chain? We're talking in a beta globulin. In a beta globulin chain, we have CTC. Okay, and then the CTC will give what? Opposite. No, in RNA sequence, that is GAG. So whenever there is GAG, you will get what? You will get glutamic acid. This is normal. Whereas what is happening, instead of thymine, adenine gets replaced. Instead of CTC, you have CAC. When the CTC becomes CAC, then the counter, that means, see, the DNA has CAC, guys. What will be the RNA? The RNA will be GTG, right? GTG. What does GTG code for? GTG code for valine. Okay. So what should be the original one? Original should be GAG. Correct? No, GAG. Instead of GAG, you get GTG. If you have GAG, you will get glutamic acid. But it is not GAG anymore. Why? Because it is replaced here. Instead of CTC, it became CAC. So the product RNA will be what? GTG. So GTG will result into what? Valine. It's just one amino acid. What much can it differ? See, guys, a protein is a structural molecule. You understand this point? So when the structure of a protein changes, the function completely gets destroyed, or it gets increased, or it gets wasted, something like this. Okay, either it is a gain of function or a loss of function. That is called as a mutation. That is what mutation leads to. So here what happened is, you have GTG or GUG. Oh, yeah. See here, GUG, guys. This is, thank you for correcting me. This is the RNA sequence, GUG. Sorry. This is, thank you. Okay, for letting me know. I just slipped it. Okay, GUG. Okay, GUG. Yeah. So what happens here, whenever you have a replacement of a particular amino acid, okay, it is a mutation. The sequence has changed. So here only one amino acid sequence has changed, or one amino acid is being replaced. So that will lead to what? It is called as a point mutation. So there are two ways in which you can define the disorder of sickle cell anemia. One is, it is a point mutation, and it is also called as lethal gene. Lethal gene. Okay. And this is a gene, okay, that affects multiple phenotypic characters, pleiotropism. You can see that a person having a sickle cell anemia will have poor development of the spleen. You can see that they have a poor metabolic functions. You can see that they are always gasping for their breaths, or they have excessive tiredness, okay, and they show weakness, excessive weakness, okay, or mountain sickness kind of a condition they can show. So it is a pleiotropism. So each gene can control many phenotypic expression. So when you talk about sickle cell anemia, you can talk about in three sense. What are those? One is, it is an example for point mutation. Sorry. It is an example for, it is an example for point mutation. Second one, it is an example for lethal gene. Third one, it is also an example for pleiotropism. Okay, got my point, guys? So what is happening here? At the sixth position in the beta globulin chain, an amino acid sequence get changed. So instead of glutamic acid, what do we have? It is replaced by valine. So what does it lead to? This leads to the deficiency of the protein. Okay, what deficiency? The work deficiency. It is not able to do something. What is it not able to do? Pick up sufficient amount of oxygen molecules. So when it cannot pick up sufficient amount of oxygen molecule, that leads to a condition called as low oxygen tension. So that leads to a low oxygen tension. Whenever there is a low oxygen tension in the shape of within the RBC molecule, red blood cells molecule, so that leads to, okay, the change in the shape of the red blood cell from biconcave to the sickle cell. So that is what leads to sickle cell anemia. So what happens in anemic condition, guys? There is no sufficient supply of oxygen to the tissues. Now you know what happens when you don't have sufficient oxygen supply to the tissues. Less energy, low metabolism, poor development of organs, etc., etc. Okay, so this is called as sickle cell anemia. Anybody has any doubts? Yeah, guys. Autosomal recessive. Yeah, autosomal recessive. That's correct. Okay. Sham, thank you for correcting. Okay. And Sultana. Okay. Jabra Sultana. Let's go with thalassemia. Okay. We have a little bit time. Okay. In thalassemia, what happens? Thallus anemia. Okay. Here also we have the same situation. Thallus anemia can be of two types. What is that? Thalassemia. Again, anemic condition. Here we have a whole defect in the whole chain, not just point mutation. We have a bigger mutation compared to that of the sickle cell anemia. We have two types of thalassemia. One is alpha thalassemia, other one is called as a beta thalassemia. In alpha thalassemia, the protein, the gene responsible for production of alpha chain is defective. Whereas in beta thalassemia, we have the beta chain that is defective. So what happens here, guys? One is the change in the red blood cell shape. Okay, the red blood cells are pretty small. The lifespan of the red blood cells is very abruptly reduced from 120 days to 90 days. Okay, so this change in the shape and the reduction in the lifespan of the red blood cells again leads to what? Anemic conditions. Usually the number of red blood cells within the person who is suffering from thalassemia is far less compared to that of a normal person. So what kind of a disease it is? Again, it is autosomal recessive disorder that is very, very similar to that of sickle cell anemia. Here it is not a point mutation. It is the defect of a whole sequence of a genome which is responsible for the production of either alpha globulin chain or it is beta globulin chain. So that leads to thalassemia condition. Okay. Is it clear, guys? Okay. What about phenylketonuria? What kind of a disease is phenylketonuria? What is happening in phenylketonuria? That should be our question. Sorry. Yeah. What is happening in phenylketonuria? So basically what happens is I'm running out of time. That's why I have to hurry a little bit. Okay. Anyways, now you guys tell me what should happen in phenylketonuria? See, we have a normal metabolic condition, metabolic situation where you have phenylalanine. The phenylalanine should be converted into phenyl hydroxylase. Okay. So this phenyl hydroxylase is an enzyme. Okay. Phenylalanine hydroxylase is an enzyme. So imagine that this enzyme is not manufactured. So if this enzyme is not manufactured, it leads to something called as phenylketonuria. So what is the function of this enzyme? So whenever there is phenylalanine, the phenylalanine has to be converted into tyrosine. Okay. And the tyrosine will eventually become what? Melanin. That is the pigment, melanin pigmentation. Okay. So, but this process is abrupted. How? In the condition of phenylketonuria, the gene responsible for production of this enzyme, that is phenylalanine hydroxylase, is defective. So that happens what? It leads to the breaking in conversion of BH4 to QBH2. Anyways, you don't need this. When this does not happen, the entire process get abrupted. Okay. So what happens here? So when the accumulation of phenylalanine, you don't have the enzyme. When you don't have the enzyme, this entire process gets what? Cancelled out. When this process gets cancelled out, what happens to the phenylalanine? You have the phenylalanine. The phenylalanine is converted into phenylpyruvate. And finally, it leads to the formation of phenyl acetate or yeah, it leads to the formation of phenyl acetate. So when you have the accumulation of phenyl keto bodies within the brain cells, now it leads to a lot of defects within the body. Again, this is also a pleiotropic condition where one gene controls the phenotypic expressions of different parts of the body. So the kids who suffer from phenyl ketonuria, what happens here is they have a very, very poor development of the brain. So because of this, they have a retarded condition, learning disabilities, motor disabilities. That means they cannot walk, talk, or function normally. Okay. And poor development of the limbs. Okay. And then they have the open mouth where they will be having the dripping of saliva. They cannot speak. Their speech is defective. So these are some of the symptoms of phenyl uh, ketonuria. Hair falling. Okay. Poor lower portion is not seen. Okay, lower it is phenyl acetate and okay, phenyl acetate. That is the thing. Okay. Okay. Anything else? I have shared the PPT. Please just go through it. Okay. Chalo. We'll stop here, guys. Let me not rush through. Okay. I hope this topic is also clear. Can you tell me what kind of a disease is your phenylketonuria? What kind of a disease is phenylketonuria, guys? Are you there? Oh, don't leave the session already. Please tell me what kind of a disease is phenylketonuria? You can unmute, you can tell me, you can on your camera. Okay, we are in the closed classroom. Okay, you can do everything. Not everything means you can converse with me freely because it's better. Okay, autosomal recessive. Again, that's correct. Okay, thank you, guys. It's an autosomal recessive disorder. That means it will have the similar functions. Okay, a similar way of expression compared to that of your which disease? Your sickle cell anemia, your thalassemia, etc. Okay. Yes, yes, yes. It's a recessive disorder. Correct, guys. Correct. Yes. Thank you so much for attending the session. It was an excellent strength. I would like to see you more of you guys. Come, please come to the session. We'll have more productive discussions. Okay. I promise. I will not waste your time. Okay, and the next session, I'll give you more assignments on solving the questions. Please keep the track on it. Okay, if you come to sessions, you'll get the assignments. And that means you don't skip the sessions because I give the assignments. Please solve and then try to give me the feedback. So this is the questions I cannot solve. Then I can really answer in the next coming sessions. Sir, where can we get the recorded classes? Guys, it's in the YouTube. I went live on the YouTube. You can go for Avanti Live 8 or Live 4. There is English and Hindi classes. Whichever the language you're compatible with, please go there, check out the classes, whichever the portions you skipped, there you can get the proper video. Okay. If you cannot see the board, you can see very well in the YouTube, I think. Okay. Yes, yes. Thank you, guys. Have a nice day.